Hi all, thank you to join this sub-module on the biological embedding of social determinants. For this module, I would like you to be able to, as a learning objective, I would like you to be able to explain how social disadvantage can become biologically embedded through physiological mechanisms, such as stress response, inflammation, and biological aging. Describe the importance of sensitive periods, particularly in early life, in shaping long-term health outcomes, and then apply the life course perspective that we have mentioned already in the previous submodule to analyze how social experiences contribute to cumulative biological risk and health inequalities across the lifespan. So let's start again with the mechanisms explaining the link between socioeconomic conditions or social adversity and health outcomes. As we know, the link between these conditions and health can be either direct or as mediated by several mechanisms mentioned earlier. So I will go quickly here. They are generally taught as environmental exposures, behavior exposures, psychosocial exposures, and healthcare access and use. So for all of these mechanisms and socioeconomic conditions to have an impact on health outcomes and disparities in health outcomes, they have to be somehow biologically incorporated. And this is what we are going to discuss today. But first, let's go back again still also to the life course approach. It is very important to frame the biological incorporation of social exposures into the life course framework. You remember we have discussed this, how across the life course, exposure to socioeconomic conditions, either on their own or as mediated by all of these factors, translate into different trajectories of health and decline depending for different individuals. And this originates in differences in health and aging. An example of how the link between an example of how we can see the link between life course socioeconomic conditions and health outcomes comes from this study that is based on the British Whitehall 2 cohort, and we use two measures, types of measures usually when we measure lifetime socioeconomic conditions. One is called cumulative life course socioeconomic conditions, and it's just a measure of how many times a participant or someone has been exposed to adverse socioeconomic conditions across their lifetime. So we measure, for example, in childhood, middle age, or an old age, and we measure a cumulative exposure to these adverse socioeconomic conditions. And another is called life course social mobility. And this one is more of a measure of the social trajectory of an individual. So, for example, in this study, we would divide into someone who has been in a high socioeconomic position in childhood and in adulthood, someone who's been in a low socioeconomic condition across their lifetime, or someone who had a downward or upward mobility. And as we can see in this graph, in this time looking at type 2 diabetes incidence, for both measure, for example, the higher someone had been in a low socioeconomic condition, the higher was the incidence of type 2 diabetes, or here looking at social mobility, those who had a low socioeconomic condition, both in early life and later life, had a higher incidence of type 2 diabetes. Early life is particularly important in this setting, and as you remember in the previous submodule, we discussed about the different ways through which lifetime socioeconomic conditions are expressed in later diseases, and one was the sensitive period model, and this is just it takes back to the sensitive period model, but shows how adverse early experiences, such as child neglect, adverse socioeconomic conditions, family violence, poverty, or other exposures like structural racism, can become biologically embedded, again, especially in sensitive periods, and especially in early childhood. These early stressors influence biological systems in several ways. They can alter brain structure and function, dysregulate the stress response system, weaken immune functioning, and lead to long-term metabolic changes. Epigenetic modifications such as changing in DNA, methylation, or chromatin remodeling are also some of the key mechanisms linking early environment to later health. And we will discuss about this a little bit more later. And then, of course, these biological changes don't occur in isolation, but they are really, they shape behaviors, increase vulnerability to later exposures, and then contribute to developmental trajectories that themselves contribute to social and health disadvantage later in life. Over time, this can lead to a higher risk of adult outcomes and chronic diseases. In terms of the link between socioeconomic conditions or social adversity and biological markers, this table represents all of the systems in which an association has been found, which is practically almost all the systems. That said, in practice, research has really concentrated around some of them that are the HPA axis, and then brain structure, inflammation and the immune system, or genome regulation, such as epigenetics or gene expression. If we go back to the immune system and inflammation, this study again on the YTOL2 cohort shows the link between social mobility, that is the social trajectory measure that I just mentioned for type 2 diabetes, and the blood concentration of two inflammatory markers, in this case CRP and IL-6. And as we can see here, we see an almost dose response association between going from the lower the social mobility trajectory, the higher the concentration of these markers. The study, this following study, based on the EPIC Italy data, also looks at the social mobility trajectory, but in this case, in relation to the DNA methylation of three pro-inflammatory genes. And as we can see here, again, the lower the lifetime socioeconomic conditions, the higher the methylation of these pro-inflammatory genes, which generally corresponds to a higher expression of these genes, which would translate to a higher circulation of these pro-inflammatory molecules. In this study, unfortunately, we didn't have information on gene expression and circulating inflammatory markers. In this other study, we used yet another measure of biological age or biological aging, and it is still based on DNA methylation, but in this case, as you can see, it's two scores, and the scores are indexes of epigenetic age. So these scores have been developed by others on. And they are generally developed with the intent of predicting morbidity and mortality, and in relation to chronological age. And for these two indexes of what we would call epigenetic age, if we go again to the right-hand side, we see this measure that we are following of life course socioeconomic conditions as measured by social mobility. And as we can see, the lower this life course socioeconomic condition measures, like here individuals who were stable low, the higher the epigenetic age of these participants for both of these epigenetic age markers. In this following study, which is based on the Swiss COLOS study, we used yet another measure of biological age, which is called pace of aging. This is an index that is calculated based on several biomarkers in different systems. And we used here again measures of life of socioeconomic conditions in adulthood, but also again the measure it's on the bottom and the right hand side of social mobility. And we can observe here again that individuals with a stable low socioeconomic trajectory had a accelerated aging as based on these markers. We have focused a lot on socioeconomic conditions, but as we know, the social determinants of health comprises a large number of, of markers and measures, and this is an example of how race is also related to biological age. And the weathering hypothesis actually was first conceptualized by Geronimus, thinking about the fact that chronic exposure to social adversity, in this case, including racism, leads to early health deterioration. And this study, which is based on the enhanced data, shows the trajectories over time of allostatic load, or the probability of having high allostatic load. Allostatic load is another measure of biological age, which is based on the physiological dysregulation across multiple systems. And as we can see here, we see Black women and white women of both high and low socioeconomic conditions, and the key findings of this study was that for Black women, either of high or low socioeconomic condition, almost at any time, this measure of biological age was higher than for white women, so independently of the socioeconomic circumstances. So in this case, socioeconomic advantage did not protect Black women from the accelerated aging in relation to their race. Racism is then one of the most important chronic stressors, and these are an example of a few studies showing, for example, telomere shortening among African American men experiencing racial discrimination, or epigenetic age acceleration among epigenetic age acceleration among African American women that had perceived discrimination. So there is also a biological embedding of chronic discrimination that affects the HPA axis, inflammation, and cellular aging. The research on the biological incorporation of social exposures actually first originated in animals, as you can imagine, and I wanted to present one of the seminal studies in this field. This study was conducted among rhesus macaques, and they examined dominance rank, which is in macaques a sort of a proxy of socioeconomic status in humans. And they were able to show that the lower the dominance rank that you can see in the figure as higher numbers, the higher the expression of several pro-inflammatory genes. But what was really remarkable in this study, also due to the experimental setting, is that they were able to show that by changing the position of these macaques across the different cages, the expression of these inflammatory genes also changed, and they were actually able to predict the dominance rank based on the expression of these pro-inflammatory genes. So this is very important because it suggests that potentially changes in socioeconomic circumstances can also then sort of buffer some of the biological embedding of these exposures earlier in time. All of this research, in all of this, is also very important to mention the conserved transcriptional response to adversity model, and it is more of an explanation or sort of hypothesis of how this social experience, how and why these social experiences actually translate into these biological differences at the gene regulation levels. So we know that when we encounter stressful or threatening situations, whether it's discrimination, social exclusion, social adversity, disadvantage, the brain doesn't just interpret these signals, but it initiates a biological response. This begins with a social signal transduction, and then the brain, and more specifically the social brain, evaluates whether the environment feels socially safe or threatening. And if the environment is perceived by the brain as threatening, it activates several biological pathways. First, the sympathetic nervous system ramps up and releases norepinephrine, which travel through the body and interact with receptors on immune cells. This shifts the gene expression to favor inflammation, essentially priming the body for injury or attack. But then the HPA axis also start being activated and releases cortisol, which normally works to dampen inflammation, but under chronic stress, the body can develop resistance to cortisol, allowing the inflammation to continue unchecked. And then, after many other regulatory systems, we can expect to have an impact at the genomic level. And how does this happen? At the genomic level, the stress response pathways actually change the way genes are expressed, as we have mentioned. And so the SNS through norepinephrine reduces the expression of antiviral genes, and then while increasing the expression of pro-inflammatory genes. And this pattern of gene activity is what we call the CT array profile. And in the short term, this kind of shift may be adaptive, as it prepares the body to attack an injury or infection, which may have been useful in other circumstances, in the past, in our evolutionary history. But then when this stress is chronic, as it is often the case in the case of social adversity, these inflammatory pathways never switch off, and over time they contribute to the development of chronic conditions related to increased inflammation, such as cardiovascular disease, depression, metabolic dysfunction, and the accelerated biological aging that we have seen earlier. And then again, going back to our original figure showing three hypothetical individuals, over time, the exposure to all of these, the socioeconomic disadvantage, the social adversity, racism, discrimination, and the associated exposure leads to differences between chronological and biological age that are then different between individuals. This field of research has several challenges. The major one is the lack of studies with very good biomarkers that are needed to look more precisely at the impact of socioeconomic conditions and the social adversity and their associated exposures. And even fewer are the studies with repeated measurements of biomarkers over time. A second major challenge is the lack and the difficulty of tracing exposure since conception and across the lifetime. There are very few birth cohorts with long follow-up and with biomarkers. Then the effect of social adversity on these biological systems and biomarkers is generally moderate. However, first of all, we only measure biomarkers in certain systems and with different scales and looking at different specific biomarkers. And then also it is to be noted that the most disadvantaged parts of our society are generally not participating to the studies that we use to look at the biological incorporation of social experiences. Because of this, the impact of this research on health policies and the clinical applications are uncertain, and of course, and I think much more translational research is needed in this field. I would also mention that this type of research requires an extreme need for multidisciplinarity, which of course represents a challenge. About perspectives in this area of research, One part I'm particularly interested in, and there is a lot of research still to be done, is around where do aging trajectories start to diverge. There are several elements that make us think that since very early in life, socioeconomic conditions are having an impact. An example is that at the biological level, lower parental socioeconomic status has been related to shorter telomere length and to higher CRP concentrations. There is also evidence to suggest that premature babies have an accelerated senescence of cord blood progenitor cells. And we know that adverse socioeconomic conditions increase the risk of preterm birth. So the idea would be, going back to our figure, to look at exactly when these trajectories may start to diverge, and then more specifically to look at the mechanisms would be particularly important. And then again, another very important part that is still under studies is around protective factors that can buffer some of the impact of early life exposures into on later diseases. And some examples here are studies looking at the maternal warmth that can buffer the effect of low socioeconomic conditions in early life, or psychological and biological resilience. Or, for example, in another study published in The Lancet, health behaviors and lifestyle can actually help in buffering the impact of adverse socioeconomic conditions from early in life. So I thank you very much for being patient and listening to this module.